All right, we are now at lecture 6. Okay, so in this lecture, we're going to cover the directionality of nucleic acids. We're going to get right into the secondary structure of DNA. And we're going to talk a little bit about Hershey-Chase and Avery experiments. So those are the two most famous experiments regarding the identification of DNA as the genetic information. Okay, and by genetic meaning like how traits are passed on. Okay. All right. So before we move on, I still want to get back to the idea of the formation of nucleotide or polynucleotide, especially in the phosphodiester bond. Okay. Still want to talk a little bit about that, right? Because I want to make sure you guys really understand what I mean. Okay. So again, the direct addition of nucleotides to growing chain of DNA or RNA is really thermodynamically unfavorable. And what that means is essentially, if you look at the slide up there, this is saying if you take a growing chain, okay, if you take a growing chain and you add to it a nucleoside monophosphate, okay, if you want to directly add a monophosphate, then the energy is going to be plus 25. Okay, so actually it favors the reverse direction. Okay, so in essence, you want to break DNA rather than make it because the energy here is positive 25. Okay, so turns out in evolution or just in biology, we are going to use nucleotide triphosphate as the building block instead of the monophosphate. Because remember, we talked about this in the very first class or maybe the second class about how if you have 3 phosphates, you got like a lot of negative charges. And if you hydrolyze the phosphate, if you hydrolyze it, then you provide a large amount of energy. Okay, so the hydrolysis of your triphosphates will generate you energy. Okay, so this is listed right here. Okay, so if you were to take a nucleoside triphosphate, okay, like for example ATP, you add water to it and then you cleave it using water. Okay, so you got water coming in and this breaking will generate you like minus 31 kilojoules per mole. Right, so you remember energies are additive, so you add them up and then you get a negative 6 kilojoules per mole. So then the DNA polymerization has just become favorable. Okay, so this is sort of one of the bottom lines that I want you guys to be sure you guys know, which is in reality, DNA are made from DATP, DCTP, DTTP, and DGTP. Okay, so they're made from nucleotide triphosphates instead of the monophosphate. Okay, so this actually has an implication in a cell's energy. Okay, so you guys know that ATP is like the cellular energy that we use to power moving, to power chemical reactions. So ATP obviously is going to relate it to RNA polymerization. ATP is going to relate it to DATP, for example. Okay, so what that tells you is that, you know, when your cell divides, when it's making DNA, your cell has to have energy. Okay, and that energy is going to come from ATP, and ATP is going to come from stuff that you eat. Okay, so we'll talk about metabolism next semester, but for this semester, we just focused on the structures and the nomenclatures. Nucleic acids are also called metastable. Meta meaning kind of, okay, like half in the middle, metastable. Metastable compounds are really, they are thermodynamically favored to break down. And why is that? Because if you look at essentially this equation right here on the top, positive 25 in the polymerization direction, that means it's going to be negative 25 in the breaking direction. Okay? So DNA is actually favored, thermodynamically speaking, in breaking. Okay? So this is why if you put DNA in solution on room temperature, if you incubate it in room temperature for a long time, eventually your DNA will get hydrolyzed, okay, eventually. Right? But it's called metastable because it's relatively stable because the activation energy for breaking it is too high. Okay? So even though it's thermodynamically favored to break down, but do so only very slowly, okay, unless the reaction is catalyzed by an enzyme called nuclease, okay, or if you add acid or base into the solution, okay, like unless you speed it up, otherwise DNA is quite stable, okay, but eventually they still degrade, okay, that's DNA, okay. RNA of course degrade faster, okay, because that extra hydroxyl group makes it easier to pop, and in addition it's a single stranded usually. Now this is just a pictorial view of what I just showed you in the previous slide, where if you take, this is the growing chain, and if you add a monophosphate, for example, this is going to be the NMP monophosphate, then you see how the arrow for dehydration is very small, and there's like a huge arrow for hydrolysis. So it tells you that your DNA wants to go back. Okay, so this is just a hypothetical dehydration reaction. Now, the reaction really favors the hydrolysis of the poly. Sorry, not peptide, nucleotide. So polynucleotides are actually formed by using the triphosphate, like what I just said. And when it undergoes the reaction with the growing chain, you're going to cut off this bond right here, and you liberate pyrophosphate. Pyrophosphate is essentially just two phosphate glued together into a phosphoryl anhydride. And this is going to drive the reaction downwards. Okay, this is not going to be reversible because you're not going to use DNA to make ATP. Okay, like, yeah, that doesn't happen. Okay, so the enzyme, okay, so this is actually done by an enzyme, and this enzyme is called polymerase. Okay, so you got DNA polymerase, you got RNA polymerase, okay, you got like whatever polymerase. Okay, so there are different types of DNA polymerases, different types of RNA polymerases inside of you, but in general, those are the guys that going to break the bond on your triphosphates and then add the base onto here. Okay, so notice how we are going to form a new bond for this 3' hydroxyl, and then this hydroxyl group on the 3' carbon, this thing, and then you're going to form a new bond between here, essentially, okay, and you generate, this guy is a 5', so you will have a 5' to then this guy will be a 3', so you keep on doing 5' to 3' polymerization. All right, the sugar positions, please, you guys have to know that. Okay, so you gotta know that 5' carbon, 3' carbon, okay, but we will not talk about like the numbering until towards the end of the semester when we talk about sugar chemistries. Okay, so for now, we're good with just the numbering. Right? You have to know this. So as a result of the polymerization, it turns out that DNA will have directionality. Okay, so the phosphodiester bond or phosphodiester linkage between the monomer units is between the carbon of one monomer and the carbon of the next. Thus, the two ends of a linear polynucleotide chain are distinguishable. Okay, so you can tell which one is like the head and which one is the tail. So one end normally carries an unreacted phosphate. Okay, obviously that's going to be the 5' end. And then the other end will have an unreacted hydroxyl group. Obviously this is going to be the 3' end. Okay? And a polynucleotide chain has individuality determined by the sequence of its bases, because every monomer of your DNA could be a choice of A, T, C, or G. Okay, so that's the individuality, right? Because like you have a 10-mer DNA, okay, all A's is different from all G's, right? So they're different things, okay? And this is, in general, we call them the nucleotide sequence, okay? And this sequence, we also sometimes refer to that as the primary structure of your nucleic acid, okay? So the primary structure for nucleic acids is nothing more than just the sequence of the nucleic acid, okay? And a while back, people used to represent the bonding like this, where you see how this is on the top, you have the bases, like A, C, G, T, T. These are the bases. And essentially, here you have, that's kind of like the sugar. And this is the 5' end. And this is the 3' end. So as you can see, on the 5' end, it's always attached to a phosphate. And on the 3' end, it's always attached to unreacted OH minus. Okay, so people, you can choose to represent DNA this way. And what it sort of also shows you is there's a phosphate in the middle between every nucleotide. Okay, so this notion or this notation shows that the sequence of nucleotides by their letter abbreviation ACGT. This notation also shows that there's the phosphodiester links are between hydroxyls and phosphates. Okay, why is that? Because 3' here is a hydroxyl group, 5' here is a phosphate. So when the 3' end links with the 5' end of the next nucleotide, it's going to be linking the hydroxyl and the phosphate. Okay? This notation also shows this particular molecule has a phosphate group at its 5' end and an unreacted hydroxyl group at its 3' end. Okay, so this notation actually shows a couple of things. And this notation also shows that it's a DNA sequence rather than RNA, okay, because it has T, not U, okay, not your cell. Right? Cool? And of course, the primary structure of nucleic acids is nearly always written from the 5' end to the 3' end. Okay? So you got other shorthand notations like phosphate, adenine, phosphate, cytosine, phosphate, guanine, phosphate, thymine. Okay? That's annoying. Okay, so a lot of people would just say A, C, G, T, T. So most of the times, this is what you will see. But for you, you should always remember the base on the left is 5' end. The base on the right is the 3' end. Okay, so it's almost always written this way. Okay, so if you see a string of ATCGs, now you will say to yourself, the first one, the one on the left, that's going to be the 5' end. Okay, and then obviously the one on the right will be the 3' end. Okay, so the main importance of DNA sequence is actually that the genetic information is stored in the primary structure of DNA. Okay, so the genetic information is stored in the sequence. Okay, it's not going to be stored in secondary structures, but rather the primary structure. Right. So for your purpose, a gene, a lot of times you hear about on the news, you're going to say, scientists discovered that a gene is responsible for cancer or whatever, things like that. Gene, okay, the word gene is nothing more than a particular sequence, okay? And by sequence, I just mean like hundreds of nucleotides together. So that's a sequence, right? A gene is nothing more than a particular DNA sequence that encodes information in a 4-letter language, okay, where each letter is one of the bases. Okay, so obviously you can imagine how a gene could have different sizes, okay, some big genes, some are small genes, but every gene will be nothing more than just a sequence made out of ATCG, okay? So it's not like magic stuff. All right, so now we're going to get into secondary structures. So long ago, ATCGs, they have this thing called base pairing, okay? And AT, A to T, C to G, they are the base pair in the Watson-Crick model of DNA. Okay, so in the 1950s, then, they were the ones that solved it. Of course, they had a lot of help from scientists all over the world, okay, a lot of data that they didn't generate by themselves. They actually talked to a lot of people. So you'll get to have a better story of DNA structure when you take molecular genetics, okay? But for now, my purpose for you is just to sort of know the chemical aspects and just to know the chemical structures, okay? So A pairs up with T because you got 2 hydrogen bonding. C and G pair together because you got 3 hydrogen bonding. Okay, now, remember I said you don't have to remember the structure, the actual, you don't have to memorize the actual structure of the DNA bases, but I did want you to remember the functions, or at least the characteristics of the nucleotide bases. That is, they have hydrogen donors and hydrogen acceptors. Okay? For example, if you take a look at this guy, this is a keto group, and what you should notice is that the keto oxygen here, that's going to be a hydrogen bond acceptor because it has lone pair electrons, okay? And next to it, you got the adenine, which is like that, and this guy is going to be hydrogen bond donor. So even though you don't have to memorize the structures, okay, because as long as you can find it when you need it, but you should be able to recognize where are the hydrogen bond donors and where are the hydrogen bond acceptors. All right, essentially this complementary system, okay, can sort of explain how genetic materials can be replicated, because based on, let's say you have A, T, C, G, this sequence right here, then their complementary base pairs will exactly fill in by themselves. Okay, so then based on this guy, you can then make DNA again, and then this is how DNA can be replicated, okay, because it's going to be a templated process. We'll talk about that in our next class. Okay, so your DNA, when it's polymerized into a polymer, and DNAs will have almost always have their complementary partners. And it turns out the way it works is that if on one chain you have 5' to 3', the other chain is going to run in reverse, okay, where you have 5' on the bottom, 3' on the top. Okay, and DNA is double helix, and helix meaning that it's going to have turns. Okay, it's going to have turns. So you have double helix, and in the middle, this is the double helix axis. So sort of you can think of it as a three-dimensional structure where it grows from this axis, okay? And every time your DNA is growing, it sort of shifts a little bit, okay? So it's actually going to shift. It's a helix, okay? Because it has rotations, right? And each base pair on a DNA double helix rotates at about 36 degrees. And actually, when we get to DNA structures, you will see how it's actually slightly off. And it depends on which form of DNA we're talking about. It's going to be different, okay? But most commonly, we observe it's going to be about 1/10 of a circle. So that gives you about roughly 36 degrees. Okay, so what that means is, for example, base 1 and base 2, they're off by 36 degrees. Okay, so this is the first base, and the second base is going to be right here. The third base is going to be right there. Fourth base is going to be right there. Okay, so it's going to be off by 36 degrees. Okay, and after it makes 10 bases, it's going to come back, and then it's going to again be at this particular position. Again, the actual scenario is going to be a little bit off, but for now, just take my word for it and say, roughly speaking, 10 turns, well, 10 bases give you a complete turn. But again, when we get to the actual structure, you'll see how it's slightly different. Okay, so 10 bases are present for each turn of the helix. So each turn meaning every time it completes a circle from the top. Okay, from the top view, every time it completes a circle, that's a turn. Okay, so that's a turn. Okay, turn. Okay, now, so DNA doesn't look like that. It's not linear. Okay, it looks like that. Okay, so DNA is double helix. So essentially, this guy right here is gonna rotate, and then after a while, you'll come back to it. Okay, then that makes one turn, right? So a turn is just a number of bases required to complete the position of that 0 base. Okay, so initially the base was here, and then 10 bases later, it comes back to the same location. Okay, so that makes a turn. Okay, because each base if you were to look at it, it's kind of flat. The bases are like conjugated. Okay, so it's like a bunch of pi electrons there. So the base is actually kind of flat in chemical structure. Okay, and because the bases are flat and they're going to be some van der Waals interaction between the bases up there. So the bases will pack very tightly together, and so there's no water inside. Okay, so it's going to pack very close. Okay, and this distance is about 0.34 nanometers, so it's actually quite small. Okay, so 3.4 angstrom. And the van der Waals keep them together, and every time you increase by one base, okay, this is called the rise of the helix. Okay, so we just talked about a turn of the helix, and this is the rise of a helix, because in a helical structure, you have also a vertical displacement. Okay, so you're always going up, right? Go up or down. Okay, so that every step between the bases, for example, this step and this step, that is the rise of the helix. Okay, so this is just some nomenclature. All right, so if we were to take a space-filling model of DNA, what you see is that you'll have a minor groove and you have a major groove. Okay, this is just a property of the double helix. Okay, because when you turn it, and these guys will always be the minor groove. Okay, so it's going to be minor, 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 and it's going to be minor on the outside, and then it's going to come back here, minor again. Okay, there's going to be a major groove, okay, between the phosphates right here. Okay, so you have a minor groove and a major groove. Minor groove is, this is sort of smaller in distance, and the major groove is larger. Okay, and if we were to take a look of the major groove, okay, yeah, actually this is not the scale of the major groove, never mind. This is the distance of one complete turn. All right, so the DNA molecule as modeled by Watson and Crick is shown here. Okay, and with each atom given its van der Waals radius. Okay, so each ball is sort of the van der Waals radius. And what you can see is that in the middle, okay, in the middle of the helix, it's very packed. Okay, it's actually very, very packed. Right? Now, the major groove, okay, this guy, it will give you more direct access to the base pairs. Okay? And minor grooves actually faces the sugar backbone. Okay, so essentially your sugar backbones face this way, and the major groove can allow you to see the bases. Right, so you got the major groove and the minor groove in your double helix DNA. Okay, so this is something you should have covered back in general biology as well. Okay, now DNA. Okay. How do we know DNA was the hereditary or the genetic information? Okay, so that's something that a while back people were wondering. You know, back then we know that life is made out of like carbohydrates, lipids, proteins, and DNA. Okay, now we know that proteins you have 20 different choices. DNA you only have 4 different choices. Okay, so a while ago, really, you have to imagine yourself as a scientist like 100 years ago, and then you're wondering, like, what kind of molecule would give you more information? One that is every residue or every monomer, you have 20 choices, or the one that only has 4 choices. Okay, so a lot of people actually thought that maybe protein was the information storage. Okay, but of course, at that time, they didn't know what nucleic acid was for. Okay, so there was a very famous experiment called Hershey-Chase. Okay, it's done by Hershey and Chase. Okay, what they essentially can show is that DNA was the thing that gets passed on rather than protein. Right? So what they started with is a virus. Okay, that has radio labels. Okay, so you know, long time ago, people really likes to use radio labels. You have phosphorus 32, sulfur 35. Okay, so they are essentially labeled components, okay? And phosphor is only found in DNA. Sulfur only found in proteins, okay? So that's how you can tell, let's say you have a protein there and it's radioactive, okay? It's going to be sulfur 35 labeled, okay? And then in addition, you can tell whether or not your thing is 32 or 35, right? So you start with a labeled phage. Phage is a bacterial virus, okay? And how do you have that? Very simple, okay? How do you get this guy in the beginning? All you have to do is culture bacteria that you're going to infect with in a medium that composes of labeled sulfur and labeled phosphor, okay? And labeled phosphor, you can have phosphates, okay? The phosphate you add to your medium is all labeled phosphates. Sulfur, in the term of sulfates, label sulfates, okay? So you can get that, right? So you first generate the phage from a labeled bacteria, okay? And your phage infects the bacteria, kills the bacteria, and you got a lot of these labeled phages. Cool? Then you're going to isolate these labeled phages, and you're going to infect a new batch of bacteria, and these bacteria are grown using the normal medium, okay? So you take the phage, and you let them infect the bacteria, okay? You let them infect them, and then you can wash off the phage particles, okay? So we know phage, this guy, protein, and whatever inside is DNA. Wash it off, okay? And now your bacteria will be infected with phage DNA, and it's going to let it grow, 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 and then when it breaks open, okay, so when the phage multiplies, okay, to a point where it's going to enter the lysis cycle, and it kills the bacteria, and you generate new phage, okay? Then all you have to do is analyze and say, hey, do I have labeled phosphates or do I have labeled sulfur? Okay? If I have labeled phosphor, then it tells me that DNA persisted. If I have labeled sulfur, it tells me protein persisted. Okay? So their result shows only you have the labeled phosphor. Tells you that DNA was the one that survived or that went through this entire phage amplification procedure. Okay, so you went from one phage to a lot of phage babies. Okay, so it tells you that it was the phosphor, which was the DNA. It was the DNA that gave you all these phage babies. Okay, so okay, that sort of says DNA is the genetic information carrier. Okay, so this was the Hershey-Chase experiment. All right, the next experiment was Avery experiment. Right, so what they were doing was, okay, they know that a certain gene or a certain piece of information or certain something, okay, inside of a bacteria will make this bacteria pathogenic. Okay, it will make this bacteria toxic to animals. Okay, but they don't know what it is. Okay, so you first isolate the pathogenic cell, okay, the pathogenic bacteria, and we're going to call that the S cell. Okay, smooth. Okay, smooth is actually named after the phenotype that they display. Okay, but you got the pathogenic cell. Right. Then what we're going to do is we're going to kill this bacteria. We're going to break this bacteria. In this case, they use heat to kill this bacteria, and you got, the cell opens and then DNA comes out. Okay, and DNA comes out in fragments because heat will actually denature your DNA. Okay, you killed the cell using heat. Okay, now the cell is broken. It has separated into its DNA and its proteins. Okay. Now, you already killed the cell, right? You killed the cell. And then you take this lysate that has DNA fragments and you incubate okay, you take a healthy, normal, non-pathogenic cell of the same bacteria, okay, a normal bacteria, instead of the pathogenic one, you mix them together, okay, you mix them together, and this gene that carries the pathogenic characteristic of this particular bacteria goes into your normal cell, okay, your R cell, and essentially what this guy can do is it's going to turn your R cell into an S cell. Okay, so it shows you that the bacterial DNA changed your bacteria phenotype. So then it again sort of shows you how DNA was sort of a information carrier. Right? So these two things sort of shows that DNA was the genetic information carrier rather than like proteins. Okay. Now it really later on it wasn't until the Watson-Crick found out how you have a genetic information relay. People were quite surprised that DNA, you only have 4 possible choices for each monomer, could be the one that carries information. Okay, all right. So the conclusion is that Avery et al. They actually showed that the non-pathogenic, this is Fumokai, so this pathogenic bacteria could be made into a pathogenic strain by the transfer of DNA from the pathogenic strain. Right? So these are the 2 really famous experiments that sort of said, okay, DNA was the one that will carry genetic information rather than proteins. Okay?